This is Fun with Micro.
The whole universe was in a hot, dense state that nearly 14 billion years ago expansion started. Wait, the earth began to cool, the autotrophs began to drool, Neanderthals developed tools, we built a wall. We built the pyramids, math, science, history, unraveling the mystery that all started with a big...
Hi, I'm David van Wyk. And I'm Stefan Visser, and this is Fun with Micro. In last week's episode, we covered a topic called biofilms.
Just a quick recap on what biofilms are. Biofilms are organized microbial communities encased in extracellular polymeric substances and associated with surfaces often of complex structural and functional characteristics.
Before I get started, I'd just like to announce last week's winner of Draw Your Own Micro competition. But unfortunately, I can't do that because there was only one entry from Yoda 31, and I know there was a JPEG of your buttocks. In this week's episode, we're covering a topic called quorum sensing.
To define quorum sensing is a process in which microbes monitor cellular density and regulate expression of density-dependent genes by sensing signal molecules released by microorganisms.
In modern terms, quorum refers to the minimum number of members in an organization.
Just to get a little bit of background on quorum sensing, the first example of quorum sensing was observed in the gram-positive bacterium known as Saccharomyces pneumoniae, though it was not yet termed quorum sensing.
This first time it was called quorum sensing was during a study of a different system in the marine luminescent bacterium called Vibrio fischeri.
The bacterium can be found in the light of certain fish and squid species.
High five! Woo. I hope you enjoyed that short break. We are now going to continue our episode on quorum sensing. Cell-to-cell communication among bacteria cells occur by exchange of a small molecule often referred to as signals or signaling molecules.
Exchange of signaling molecules is an essential role in gene expression in microbial populations.
The best studied quorum sensing system is that of the marine bioluminescent bacteria we mentioned earlier in this episode called Vibrio fischeri. It only produces light at high densities.
Intercellular communication plays an important role in the regulation of gene expression, whose products are essential for the formation of virulence, symbiosis, biofilm production, plasma transfer and morphological differentiation in a wide range of bacteria.
Quorum sensing can regulate multiple genes and operons. We are now going to go into a short break, and afterwards we are going to explain quorum sensing in detail.
Welcome back from our short break. Now to continue on our episode on quorum sensing. Quorum sensing in Vibrio fischeri and many other gram-negative bacteria uses a signaling molecule N-acyl homoserine lactone, or AHL, which is indicated on the board here.
The synthesis of AHL or N-acyl homoserine lactone is catalyzed by the enzyme called AHL synthase, which is a product of the LUX-I gene. The LUX-I gene, which is indicated here on the DNA molecule of Vibrio fischeri, is subject to positive autoregulation.
Transcription of the LUX-I gene increases as the concentration of the AHL increases inside the cell. This is accomplished by the transcriptional activator LUX-R, which is only active when AHL binds to it.
Without AHL activated LUX-R gene, the LUX-I gene is transcripted at minimal levels.
AHL diffuses freely from inside the cell to the outside environment.
As the cell density increases and the concentration of AHL outside the cell becomes more than that of the inside of the cell, as you can see here.
As AHL flows back into the cell because of the increase in bacterial population and AHL concentration outside the cell, it binds to the inactivated LUX-R gene, which then turns it into the activated LUX-R gene.
The activated LUX-R can now activate high-level transcription of the LUX-I gene and the other genes whose products are needed for the bioluminescence of Vibrio fischeri, LUX-C, D, A, B, E, and G. This concludes our episode of quorum sensing. In our next episode, we will be discussing microbial metabolism. I hope that you could have learned something from this episode. Good day. Yoda 31, and you know there was JPEG of your buttocks. Catalyzed by an enzyme called AHL synthase, the product of the LUX-I gene. Transcription of the LUX was from Yoda 31, and I know there was a photo of your buttocks. In this week's
or AHL, which is indicated
Interesting fact. In modern terms, quorum refers to the number of members in an organization.
of microbial organisms.
Microbial community. It's your own microbe.
Wow, ek het gesê soos wat? Functional. Welcome back to our episode on Quorum sensing. I hope you enjoyed the break. Now to get back to the lecture. Can I say lecture?
On last week's episode,
Acyl homoserine lactone, a signaling molecule, as displayed on the board here. Fine, with microbe.
in last week's episode, we covered a topic called biofilms.
Just a quick recap on what biofilms are. Biofilms are organized microbial communities encased in extracellular monitor cell density and regulates expression of density-dependent genes by sending signal molecules released by microorganisms.
Go. Fernand kan het uitediten.
Unfortunately, I found a topic called biofilms.
Just a quick recap on what biofilms are. Ordinary substances and associated with surfaces, often with complex structure and
I hope you enjoyed that short break. We are now going to